Ladies and gentlemen, welcome your host for this event, Corey Moreau.
Welcome everyone. We're really thrilled to have you here. For those that don't know you, I'm Corey Moreau. I'm a professor at Cornell University, and I'm also the current president of the Society of Systematic Biologists. I'm really excited to have you all here, not only for the series of events we're going to have this morning, but also a series of events we're having for members and people who support systematic biology. For those of you who are members, who are interested in being members, I'd like to encourage you to please join our annual SSB membership meeting, and that will be today at 1:30 in room 524B. It's bound to be an exciting meeting. Some of the things that we're going to share are the results of a possible name change and have a lively discussion around what the implications and motivations for those questions were. We'll also talk a little bit about our ideas for vision and branding and brand development, and talk about what does it mean to be a systematic biologist in the 21st century. We'll also talk a bit about our finances, and although that might sound dry and boring, I hope you'll understand why we think it's so exciting when you see the support we do for our trainees and early career members. One of the things I'm really excited to share is we recently adopted the Global Assistance Membership Program, and what this essentially does is provides discounts or free membership for students, educators, and researchers from 152 countries and territories around the world. So if you have colleagues who are interested in systematic biology or biodiversity, please encourage them to take advantage of this opportunity and join our society. So part of our society is that we have two flagship journals. The first is the Systematic Biology Journal, which I'm sure many of you are familiar with. It's free for members to publish in, and it's very inexpensive for open access, so please consider submitting your papers there. We also have the Bulletin of SSB, which is also open access only, and it's a great opportunity to publish papers where you really want to get a wide readership to see them. Like everyone, we're on social media, so please do consider following us there. Most of our updates actually come out there much faster than they do in our emails or newsletters, so please do consider following us on social media. I think one of the things that really makes our society stand out is the large number of awards that we present. We have the Ernstmeyer Award Symposia that happened during the virtual meeting, and we'll talk a little bit about that. Recently, we started a program called the Early Systemicists, where we gave out free membership to a large number of individuals to join our society. We have the Graduate Student Research Awards, which support research by early career members of our society. The Mini Arts, which is a great program. Please do consider applying, and that's across career stages. And the Publisher's Best Paper, and lastly, the President's Lifetime Achievement Award. I really want to take a moment to highlight how much money we invest in our members, especially our early career members and trainees. Last year alone, we gave out $173,000 to our members. This is a great reason to be a member of our society. We really do invest in the next generation of systemicists and biodiversity scientists. So this year, during the virtual symposium, we had the Ernstmeyer Award Symposia. We had a large number of individuals who were qualified and selected to present in that symposia. This is the first of 2 pages of those members. If you're on this list, please stand up and receive a round of applause for your outstanding presentations during the virtual conference. I can attest, I watched every single talk online and they were fantastic. I also want to take a moment and thank the award judges. It's a lot of work to evaluate these candidates. They used a very thoughtful rubric, and I can share that I know that it was a very difficult decision. In fact, it was such a difficult decision they couldn't pick one winner. In the end, we actually have 2 winners for the Ernstmeyer Award this year. First is Herku Handike, and I know that he's not able to join us today, but please do give him a round of applause. And he's from Louisiana State University, and he presented on rethinking software development for more accessible phylogenomic research and teaching. And next we have Jerica Jameson. If you're here, please stand up and receive a round of applause. Great, thank you. And so she's from the University of Toronto, and she studied the convergence of glucose and fructose metabolism across multi-evolutions of fruit and nectar feeding habits within bats. As I mentioned, we also this year started a new program, the Early Systemicist Award, and we gave out a large number of memberships across, I'm going to show you 3 pages of names. And if any of you are in the room, please stand and receive a round of applause. Another category where we've been able to support early career members of our society are with the OUP Publishers Best Paper Awards. So in 2022, we actually highlighted 2 different papers to receive the Best Publishers Award. And in 2023, we also highlighted 2 individuals to receive the Early Publishers Best Paper Award. So if any of you from 2022 or 2023 are in the audience, please stand and receive a round of applause. And I hope all of you were able to see Dr. Corey Welsh's outstanding presentation yesterday as part of the 2023 Tri-Society Award for the IDEA Award. I know that many of you were there. I saw many of you. It was a fantastic presentation that helped us all think about how do we decrease the gap in performance and success within undergraduate education and actually make the academy a more welcoming, inclusive place. And so I really want to congratulate and thank Corey Welsh for an outstanding presentation yesterday. And for this next award, I'd actually like to invite Jessica Ware, who's our president-elect, and David Baum, who's our past president, to the stage. One of the awards that we are allowed to give is for the President's Lifetime Achievement Award. And this year, the 3 presidents have decided to award this prestigious honor to Dr. Scott Edwards. Scott, will you join us on the stage? So every 3 years, the 3 Society for Systematic Biologists presidents, the past, the present, and the future, have the option of choosing a recipient of the President's Award for Lifetime Achievement for outstanding contributions to the field of systematics. The President's Award is given to a person who has made outstanding and sustained contributions to the field over the course of many years. On behalf of myself, Corey Moreau as president, Jessica Ware as president-elect, and David Baum as past president, we want to recognize Dr. Scott Edwards with the President's Award for Lifetime Achievement. Scott's work has advanced our understanding of the use of large-scale molecular data for species delimitation, added to our understanding of how historical geography structures populations, leveraged genomic data to understand connections between genotype and phenotype, and has done all of his scientific research with an eye for diversifying science and being an excellent mentor to the next generation. And I know many people in this room can attest to that. He has published important papers in our flagship journal, Systematic Biology, and has advanced the field more generally with his empirical and theoretical studies. Scott has led efforts to provide young students with the opportunity to attend scientific conferences. The Undergraduate Diversity and Evolution program, which is running this year and has for many years, was started by Scott and colleagues. And he attends SACNAS, the Society for Advancement of Chicanos, Hispanics, and Native Americans in Science, almost every year to serve as a mentor for undergraduates from historically marginalized groups and to help them learn about scientific careers in ecology and evolution. Scott is a role model for not only how you do truly integrative research using state-of-the-art molecular tools, but also serve the broader scientific community. For these reasons, we are thrilled to bestow Dr. Scott Edwards with the Society of Systematic Biologists President Award for Lifetime Achievement. Please join me in congratulating Scott. So at this time, I'd like to give the opportunity for Scott to say a few words before we move on to the presidential's address.
Thank you so much, Corey, and thank you, David and Jessica. This is a huge honor, and it's just really great to be back in a community that has nurtured me for most of my career. I think I attended my first Tri-Society meeting back in the late 80s as a graduate student, and it's really impressive how the society has evolved both intellectually and as a community. I was very fortunate in graduate school at UC Berkeley to be mentored by people like Montgomery Slatkin, who at the time was one of the only people teaching about things like coalescent theory and ways to integrate micro and macroevolution. And for that, I'm very grateful. I also just wanted to thank a couple people who, over the years, have really helped put ideas that I've had in a concrete form in a way that I simply couldn't do myself. People like Peter Beerley at Florida State University and Liang Liu, a former postdoc now at the University of Georgia. In terms of the outreach that Corey mentioned, this has been very much a team effort. Rich Kleeman continues to run the really amazing UDE program, along with some really dedicated community members like Maria Uribe and others, all of the mentors for the students this year. This is a great community, a great tradition, and I'm just so thankful to have been able to help nurture it over the years. I also want to give a quick shout out to Sally Otto, who turned that crazy bicycle trip I did in 2020 into some cold hard cash for the Tri-Societies to nurture the next generation. So I hope this Lifetime Achievement Award doesn't mean that I have to pack it up and quit. But thank you so much, Corey and David and Jessica.
So the real star of the show today is our past president, David Baum. He's going to be presenting the presidential address. And in his presidential address, he'll be exploring some uncomfortable truths about species. So please join me in welcoming David to the podium.
Thank you. Good job leading it. Well, I want to start by saying thank you to Corey for emceeing today, and congratulations again to Scott. I also just want to take the opportunity to just point out that I've spent the last 2.5 years working with the Systematic Biology Council and Executive Committee. And for the members in the room, I want you to know you have a fantastic council. They're really, truly committed to figuring out the best ways to advance the science and support the next generation of systematic biologists. And I also perhaps should thank the members for electing me as president a few years ago, although right this minute it's a little mixed feelings about that. Because I now get the opportunity to share supposedly words of wisdom with this largest audience I've ever spoken to. So I reflected on what this means, and I realized that not everybody gets the opportunity to be given such a stage. And it led me to want to do something that I haven't seen done very often, but I think it's important to do once in a while, which is to really reflect briefly on what the factors that allow or enable or prevent us from having successful careers. So in the same spirit that we often will give a land acknowledgement nowadays, to reflect, the fact that we're privileged to live on some land, but people were thrown off that land, so our benefits come at other people's detriment, and that obliges us to do something about it. I've written for myself a career acknowledgement that I hope you'll indulge me if I could just read this out. So I've been blessed with a wonderful and fulfilling career. This is not simply the result of innate talent or hard work, but entailed abundant good luck and undeserved privilege. I acknowledge this reality and the many individuals who have unfairly missed out on opportunities that I was granted. I believe that this reality obliges me and all similar individuals to honor those who've helped us and to give back to the community through teaching, service, and especially by doing what we can to advance the careers of individuals who have had fewer privileges. So, I am a white, straight man with certain privileges, and beyond that, I was very fortunate. And just to kind of name some of that fortune, I grew up in a family surrounded by science. So my father, son of immigrants, grew up very, very poor, nonetheless was successful, but was a successful biochemist, and he's still alive, he's still with us. He and other extended family was grew up with science, living and breathing science from a very young age. And I always had this notion that I carry with me till today, that science is a grand enterprise. We're trying to answer really exciting big questions and use our creative skill sets to tackle things in new ways. He also, I think, was a model in the sense that he was committed to education and outreach. He's most famous for the Biochemist Songbook, which is all available online for free if you're interested in the songs. So that gave me an advantage that I know most people don't have. I was also very fortunate to get mentors throughout my career, as I think everybody who's successful does, who supported me in all those important ways, not just providing opportunities, funding, pastoral support, academic support, and inspiration. And I've put a few up here, the sort of major players in my undergraduate on your left, Barry Juniper and David Mabberley. At graduate school, I've picked out Peter Raven, Peter Hoke, and Alan Larson. My postdoc mentor, and now my colleague, Ken Seitzma, and Michael Donahue, who has really mentored me tremendously when I was starting out my career and as an early faculty member. Now you'll notice that all of these people, like me, are white men. I think the one thing that I have tried to do, and I think we should all aspire to do, is to make sure that our mentees are more diverse than our mentors. And I think at least I can say that's sort of true as a first sweep. I have been incredibly lucky to have worked with many fantastic students over the years. I have 3 wonderful students right now. These are the 17 students who have sort of gone out of my lab with a PhD, and it's just been a pleasure and an honor to work with them. 3 of them at least, I think, are at least in the meeting. Maybe they skipped the meeting, this talk, I don't know. But, and so that's something that I both feel proud about, but also honored to have worked with these great people. I've also had 15 postdocs in my career. It's kind of fun at this point to go back and count and then find pictures of people as they are now. I encourage you to do that once in a while. And of course, we've all benefited from uncountably many other people. Family, all the other students, undergraduates. We mustn't forget all the administrators, field assistants, technicians, and grant program officers, and all these people that make this possible. So I want to just give tribute to all those people who made my fortunate career possible, and just say that I plan to continue as best I can to pay back to the community within which I've been so fortunate. So that's just my intro. But I don't feel qualified to talk about much else except for science from here on out. But before I do, I wanted to give you a little quick map of my career trajectory, because it's not a typical trajectory, and it did pose me some problems in preparing this talk. So I think of the study of science as an investigation of the landscape of knowledge, where we're trying to scale peaks to get a better view, to understand the world the higher we get. And by that token, I think it's fair to say that I began my career in the systematics range. So I did my PhD work on the systematics and floral evolution and pollination biology of baobab trees, which I still am passionate about. And then I developed my research career in this range, studying things like phylogenetics, adaptation, trait evolution, biogeography. But, and I could maybe blame my father for this, I do have this tendency to look a little bit out into the distance. And I saw this range in the distance that I'm going to call the Evo-Devo crags. And it felt to me that there were some really interesting questions that could be addressed by climbing those mountains, understanding how it is that genetic changes change development, and those in turn change development. And so I decided to try and launch a research program in that area, which required crossing what I would call the valley of low credibility, But I had good fortune. I had a few postdocs that really helped me make that transition. And I spent, more than probably 10, 15 years really trying to understand these questions of how identify genetic underpinnings of the morphological differences between mainly relatives of Arabidopsis or closely related plant species. So that was, one sort of speciation event in my history. But after I spent a lot of time, I've done a fair bit of service at my university. So after 6 years as department chair, it was proving difficult to support the Evo Devo research. It's quite expensive. And I sort of spotted this other mountain in the distance. This is the Origins of Life Mountains. So I felt for a long time that the study of the origin of life needed more good evolutionary biology in it. And I felt like there were experiments that could be done that weren't being done that could enrich that field. So I decided, what the heck, I have tenure, maybe I can cross this plane once again. This time I did cross the plane, but I found a little holdout in the way, I'm calling the eukaryogenesis mound. And nowadays, not only have I managed to begin my exploration of this mountain range, I've actually pretty much dropped my systematics research. So my lab currently is 100% studying the origins of life, looking at, doing lots of experiments that are basically chemistry to understand how chemical systems can become organized and begin showing the dynamics we associate with evolution, and also doing theoretical work. But that poses me with a problem, because here I am speaking as past president of the Society of Systematic Biologists. I felt like, even though I might enjoy, and maybe you would enjoy, me talking about the origin of life, I felt like I should stick to the systematics range. So what to talk about? Well, if you know my history, it's sort of obvious what I was going to talk about. I was going to talk about the one peak that I have sort of felt like I've been trying to scrabble my way up almost my entire career, which I'm calling mountain species. So I don't know about you, but for me, the nature of species is a really fundamentally interesting question. As scientists, we have a sense, the way we make sense of the world is as a set of entities that sort of help us, that sort of seem causal, concrete in some way, that we use to build models. In biology, since, obviously well before Darwin and motivating Darwin, there are these units we've perceived called species. But what are they? Can we climb this mountain and figure out the best way to think about species as entities? And that's what I'm going to talk about today. And I'm going to do it in 3 sections. I'm going to first of all talk about species as taxa, which is kind of how I entered this arena. Then I'm going to shift gear and talk a little bit about species as reproductive communities. And then I'm going to end just by also touching on the question of what we should say to our students and the broader public about species. So, starting with the species as taxa view, of course, coming from the systematics perspective, this was the obvious way to think about species. Species are, after all, one of the nested series of ranks that we choose to go about naming in taxonomic systems. And it was already known, and I think it's well accepted, that the principle that guides the development of taxonomic systems is that we want to somehow reflect evolutionary history. We want the groups of organisms that we name to be united by a common evolutionary history. And so it's been established then for a long time that if we have a phylogenetic tree, that the groups we care about are monophyletic groups that are, sort of descended from a common ancestor. But this poses problems if we want to approach, think about species, because traditionally, what is that common ancestor of a clade? It's a species. So a monophyletic group is a group of organisms descending from a common ancestral species. Well, how can you put a species at the end of that without ending in a sort of circular arrangement, sort of circular reasoning? So the key insight that got me started on this field was a paper in 1990 by Kevin de Quiros and Michael Donoghue, who pointed out that monophyly is not what we actually care about. Monophyly is a proxy for what we care about. What we care about is relatedness. And it so happens that in a phylogenetic tree, all the members of a clade are more closely related to each other than to anything outside that clade. It's that property, though, that we really care about, and they called it exclusivity of relatedness. I will just call it exclusivity. So to reiterate, an exclusive group is a group whose members are all more closely related to each other than any of them are to anything outside the group. That's the criterion. And so this way of thinking raises the question, well, what is it to be exclusive for a group of organisms that, we might want to call a species? So way back when, as a graduate student in the old days, I had the pleasure of collaborating with my classmate and really great friend, Kerry Shaw, who I don't think is at the meeting, unfortunately, but to try and make sense of this. And we proposed this thing we call the genealogical species concept. We sort of proposed that we should look for exclusive groups, but base that by looking across the genome. At this point, it was known that there was a lot of discordance in the genome due to different genes having different histories. And we should take a consensus of the genes, and that would tell us which are the exclusive groups. So the groups we care about would be groups that are clades on all or nearly all the gene trees. We went one step further. This is where we went out a bit on a limb, and we proposed that if we did this, we would find that the lowest of these groups would be appropriately called species. They were the basal exclusive groups. So within species, we would not have further exclusive groups, and that would sort of provide an objective criterion for ranking species as real things. Now, this idea, it's very early. I didn't put the year on there, sorry. So I think we presented it in 1992. It got published in 1995, so a long time ago. But of course, at this time, there was a lot of coalescence theory being developed that taught us that actually genes undergo lineage sorting slowly. So if you have two groups that separate, it takes very large numbers of generations in the units of population size for them to start showing monophyly. So, if you have a population size, a typical population size of maybe, 10,000, 100,000, a million, you're looking at tens of millions or millions or hundreds of thousands of years that it will take for something like 95% of the genome to show monophyletic, to make these groups monophyletic. So that led to the way suggested that we need to think about this a little differently. Instead of expecting all the genes for a group to show a common history, to show monophyly, we should just really expect that more of the genome the group in question is monophyletic for more of the genome than any contradicting group. That for a plurality of the genome, our group should be real. And that felt to me, and still feels to me, really the essence of what it is to be a taxon. A taxon is a group that's united by a common history for at least a plurality of the genome. And so that was the criterion that I began to think about at this time. So how do we measure this? What are our metrics? Well, in 2007, I proposed this thing called a concordance factor, which I think many people have probably heard of. A concordance factor is simply the proportion of the genome for which a given group is a clade, is monophyletic. And so what we want to then ask is a given group, is its concordance factor higher than the concordance factor of any contradicting or overlapping group? If it is, we could consider it exclusive. If it isn't, we consider it non-exclusive. So this is the criterion of being a taxon that I sort of emphasized. And the question then is, well, how do you estimate this? How do you figure out what are exclusive groups and what aren't? So I'm very lucky. I have as colleagues in my department 2 statistical phylogeneticists, Cecile Lanay, who I think is here, and Brett Largett. And we sat down and I posed this question. If you were given genomic data, how would you determine the concordance factor of a given clade? And they developed this beautiful approach, a Bayesian approach, that was implemented in the program Bucky, that allows you to estimate for us, if you're given a set of genes, you can estimate, taking account of gene tree uncertainty and priors that you place on concordance, you can estimate the fraction of the genome for which a given clade is true. And you can do that in two ways. You can look at above the branches here, you see we have numbers called sample-wide concordance factors, which is just saying the fraction of the sample genes for which this clade is true, with a credibility interval. And then if we know how many genes there are in the genome, we can extrapolate using our priors to an estimate for the whole genome. And so this method is widely used. It's still actively supported. So, and it means that we can now do this, what I always wanted to do, we can now do it when we want to. And to give just one example, I picked a study from a late colleague, David Spooner, with his graduate student, Carlos Arbizu, who studied carrot phylogeny. You don't need to see the details here, but what I've done here is I've circled all the branches where the estimate of the concordance factor is above 0.5. If it's above 0.5, we know it's at least a plurality, it's actually a majority of the genome. We could be more stringent and only look at the ones that are supported with a credibility interval is entirely above 0.5. We get that, we have fewer branches with this study that have that. But I want to point out that when we do that, there isn't an obvious pattern. You don't find high exclusivity, high concordance factors, sort of towards the root of the tree, and then they get lower and lower and lower, and then they stop, and you start finding, things that you might want to call objectively ranked species. You don't find that. They're all you get high concordance factors within groups that have traditionally been called species. You also get low concordance factors for sets of species. So that is a, I'm sorry, I'm going the wrong way now. So the concordance factor is a useful metric. But to highlight the fact that despite that, there's nothing magical about the concordance factor. What we care about is exclusivity. And exclusivity, as you'll see, is not actually as clearly defined as I always thought it was. I gradually came to see that I was making assumptions by assuming that concordance is the right measure of exclusivity. So in about 2014, I was teaching a graduate class. I had this very talented graduate student in the class, a microbiologist called Eric Wright, who's now on the faculty at the University of Pittsburgh. He's also an excellent bioinformatician. And he was interested in the problem of species, and particularly in relation to bacteria and microbes. And bacteria and microbes, unlike eukaryotes, where the discordance that we see among genes is primarily due to incomplete lineage sorting or due to gene flow, introgression among closely related taxa. In bacteria, a lot of it is due to wide horizontal gene transfer, where very distantly related pieces of genome come in. And you could say, well, should we then be taking account of how distantly related the bits of the genome are to one another? Not just whether they're monophyletic, but how divergent are they? And so talking with him, I realized that you could actually define an alternative measure of exclusivity, which is almost more direct than the concordance factor approach. So for each pair of species, you can calculate for each gene a degree of relatedness, which ideally might be the time of common ancestry of that gene in those two species. Now you could do that for all genes in those two species and average it. And that will give you the average relatedness across the genome for these two species. I said species, I was stupid. These two accessions, these two tips. Now we can do that for every pair of tips in our tree, every accession. And we can then, given that, we can take a group of organisms, any group, and we can calculate something we call an exclusivity factor. So remember, low distance means high relatedness. So the exclusivity factor can be sort of understood as we identify the maximum distance within the in-group, we subtract that from the minimum distance between the in-group and the out-group, and you can standardize it if you want to have it scaled to 1. So this basically is saying if this value is positive, it's exclusive. The organisms in the group are more closely related to each other than they are to something outside the group. If it's negative, then they're not. And if they're positive, you then have a measure of how exclusive they are, if you will. And the nice thing about this is also very easy to calculate. Eric, showed this and published scripts in the paper. So this is 824 streptomycete genomes analyzed this way, where the blue branches show show exclusivity, the ones with positive exclusivity. The orange ones are non-exclusive groups, and the black ones are tips. And if you squint at that, you'll see again, there is no clear pattern. It's not like we have high exclusivity deep in the tree, and then we suddenly get down and the exclusivity disappears. And that makes sense because it turns out, as we would hope, the exclusivity factor and the concordance factors are very closely correlated. They correlate well, not perfectly, but they are very closely correlated. So this puts us in an interesting situation. I've said that I think taxa in general should be understood as exclusive groups, but I've given you 2 different ways to measure exclusivity that could differ in some cases. Additionally, and I didn't emphasize this, with each of these methods, there are decisions one would have to make. How do you measure relatedness between the genes? Do you care about the rate of evolution, or you just want to look at the time since common ancestry? Do you want to count every site in the genome equally, or do you want to count every recombination block equally? So there's some ambiguity, which is unfortunate, but I've come to realize that it's never as clean as you want it to be. Nonetheless, despite all that, that doesn't mean that exclusive taxa don't exist. It just means that depending exactly which pair of glasses we put on, we might see them appearing at a different place. There might be a few that are fuzzy, but there's undoubtedly many, many exclusive groups out there. And that's why I still believe that the field of systematics is so important. We have an obligation to go out there, identify these exclusive taxa. They are united by a common history, and figure out how to systematize them and communicate about them. And I just couldn't resist putting up this reference to this op-ed that appeared like 2 weeks ago in the New York Times, arguing once again for how important this science is. So taxa are undoubtedly real and worth studying, but what about species? Well, there's where we have a problem. As I've shown you, there is no clear rank of species based on exclusivity. There just isn't a sort of sharp transition point at which we see exclusive groups appearing, and I'm not sure why there would be. And even if we took the view that, well, we can still pick a criterion and try and just assign everything to species to exclusive groups where every organism is in an exclusive group at a certain rank, that is also impossible in practice because the world is messy. The world does not break itself up into tidy patterns. We have geography. Geography can certainly complicate things. So think about a ring species, like this one that my former student Eva Liu documented. In ring species, you can't do it. You can't do it in general for many geographically widespread taxa with complex patterns that don't allow all organisms to be placed in an exclusive but non-overlapping group. And so the bottom line is that as I have spent my career worrying about, well, what are species, I've come to this, I feel like I've sort of climbed this mountain. I wanted them to be, well, maybe not billiard balls, maybe fuzzy billiard balls, sort of, I don't know, like Nerf ball type things or something, softer but real still. And I feel like I got to the top of the mountain and I just found a blasted out crater with, no tangible, functional, purposeful species that I could really grab onto. So that, is a slightly, frustrating but exciting, because then the question is, well, what is it we're perceiving out there in the world? So this is where I want to change gear, and this is where I wanted to change gear and think about what it is that I think motivates a lot of our sense that there are these units out there that we care about. And that comes from the idea that species are not simply taxa, they are also, that word is carrying with it a role of something about the evolutionary process. Specifically, historically, we've thought of species as being reproductive communities at some scale. So let me just sort of talk this through a little bit. So what are the lineages that comprise the tree of life? Well, those lineages in sexual organisms we can understand through the lens of common descent. Not common ancestry here, but common descent. So if you imagine you could go out and you knew everything about a group of the organisms that existed, you could find a group of organisms and say, okay, each of these organisms is more likely to share a common descendant with one another than with any organism that is not a member of this group. That is a reproductive community. That's not saying that all of these organisms have 100% probability of sharing a common descendant. It's just saying it's elevated relative to the individuals that are not in the group. To me, that is what a reproductive community is. And if we take a reproductive community at a point in time, and we connect it to the following time and the one after that, we end up with a sort of tube-like thing, which is a population lineage. And so when I see what people often call a species tree, I think for obvious reasons I prefer the word population tree, drawn out, I see those orange branches as that. They are reproductive communities at a point in time, and the splitting event represents some kind of splitting of those those communities into two or potentially more at some point in time. Now in practice, it's important to notice that when we use them, when we do theory with them, we almost always make two huge additional assumptions. The first assumption is that the groups don't have substructure. They are panmictic. The second assumption we usually make is that when there's a splitting event, that's an instantaneous splitting from one panmictic population to two panmictic populations. Obviously, these are massively oversimplified, but thinking this way has really advanced the field by helping us develop the whole multi-species coalescent model that so much is that we use so much in systematics. So the question then is, what are these groups? Are they just, like I said, groups that happen to be there based on the current geography, the current biology of those organisms, or is there something real about them? Are they actually cohesive in some way? So the way that I just presented these communities is sort of saying, imagine we could state something about the likely future. So it's like looking at the present and looking into the future. So there's a very good paper that I highly recommend by Wayne Madison and Jeanette Witten in the Bulletin of the Society of SSB that came out last year, where they explore that and they suggest that, there are problems with that, because if that's all we're looking at, then they are not functional. They're just the things that happen to have, based on what they are now, they happen to be likely to share common descendants. And furthermore, how can we predict the future? So what they suggested, which I think was an interesting move, was to say that, actually, the things that are groups now, how do we recognize them? We recognize them because they are products of history of cohesion. So they basically say that we should use gene genealogies to infer the groups today, and those groups we should understand as also being predictors of the future, because there have to be some cohesive processes that result in this group being exclusive, or this bigger group being exclusive. So, so they acknowledge, of course, as I've said repeatedly, that the rank of these things is not clear. We have groups within groups. We have various levels of historical grouping, but that each of these groups can be seen as being cohesive simply because otherwise we wouldn't detect it. At least I think that's the argument. So I'm going to, I have time just to kind of explain briefly why I'm a little skeptical. At least the things I want to understand. It feels to me like we look for cohesion, but is there really cohesion? Or are we creating it in a sense to post hoc justify our perception that there are entities there? So let me talk you through a simplistic way of thinking about cohesion. And I'll just make a little side note here that this way of thinking actually is proving to be a little helpful in thinking about the origins of the first cells. Anyway, so here we have an adaptive landscape. Adaptive landscape where every point in this landscape represents some sort of ecological phenotype of an organism. And the altitude represents its fitness. Now imagine a group of organisms sitting on this adaptive peak, shown in purple. Now, those individuals will not all be right at the top of that peak, however strong selection is, because genetic drift and mutation mean that there's going to be a cloud of variation around the optimum. Well, the fact that this cloud means that if two individuals encounter one another and had some way to average or create some sort of intermediate, or in the modern world, have sex and make offspring, then that offspring will, on average, be more fit than either parent. And this is ultimately why sex has evolved, is because of this fact that given variation, and if every individual is responding to, has a history of responding to the same adaptive peak, offspring will on average be fitter than either parent. But of course, if there are other peaks in the landscape, and an individual from this purple group meets an individual from this alternative peak, well, their hybrid offspring will have lower fitness than either parent. So you would imagine selection would disfavor any variant, any sort of allele that promoted that kind of mating, and would favor ones that inhibited the probability of mating with those other individuals upon meeting. And so when we do that kind of game, we think about it, we think, well, this is what we would expect, yeah? You have two peaks, they would each have a set of organisms, and they would be reproductively isolated from each other. And I think it's a very natural way to think about it, but I've come to realize that subtly, very subtly, we are assuming that the groups exist in advance. So let me try and explain that, and this may or may not work, and you're welcome to take me to task later in the conference over this. It seems to me that if we just ask the question, take that blue individual, just point to them, well, their hybrid with this red individual would have lower fitness than that blue individual, but their hybrid with this green individual might have higher fitness certainly higher fitness than this one. So selection is not uniformly within the purple space favoring mating among all individuals. So the only reason why we would expect them to be cohesive is if, as a matter of fact, they are all breeding with one another. In fact, if it's only because the genes are flowing within this purple part that they can't find their own, they aren't going to be selected for differentially to divide. So this idea that selection will create and maintain these two boundaries doesn't seem quite right, somehow. I'll also point out that the question of how selection acts on individuals with different genotypes in terms of whether it would favor or disfavor mating between them is much more complicated than just looking at the fitness of offspring. It's also obviously going to matter what the costs and benefits are of the character change. For example, the costs of being able to distinguish, somehow between individuals situated in different places in the adaptive landscape. It's also going to matter how likely you are to actually encounter those individuals. In other words, geography becomes important. So putting this all together, I am becoming skeptical that we can explain species entities as products of selection for interbreeding potential. It just seems like we may be getting ahead of ourselves in doing this. So I want to be clear, though, I'm not saying we shouldn't study this. To me, this is one of the most exciting and important parts of all of evolutionary biology, is understanding how interbreeding potential, the propensity to mate with individuals that you encounter as a function of the phenotypes and genotypes of the individuals, that seems totally central to biology, to evolutionary biology. We need to study that and understand it. But I had this sort of intuition, concern, that maybe by intrinsically associating it with the notion of species entities, we may actually do this, we may harm the enterprise a little bit. So when I read some of the literature in this area, I picked out just 2 quotes. I intentionally didn't put any citations on here. I feel like we often slip into this reasoning where we sort of perceive that there are entities and then look at the data as somehow, we interpret what's happening through the lens of there already being entities. So I'm just throwing out there the controversial view. I figure at the start of a meeting like this, it's good to be a little controversial, that maybe we would be better off if a study of interbreeding potential were not entangled with a preconception that species entities are real functional things in the world that help us make sense of things, and other than as taxa and historical groups. So I guess what I am arguing, and as I say, provocatively perhaps, is that whether we focus on history or process, the notion of real species-ranked entities have found increasingly hard to sustain. I feel a little bit like we're chasing a rainbow, we never get there. And maybe we shouldn't be surprised, because after all, as we all know, biology is in constant flux. Evolution is an ongoing process. And if anybody here has read any process philosophy, there is this growing movement in the philosophy of science that called process philosophy, which really holds that everything is in flux, everything is flowing, and therefore entities in a naive sense don't exist. So, okay, I have kind of given my main pitch, but I want to. Okay, I'm going to dive into this. I was tempted to do a little interactive thing, but I'm going to just go straight in and just make this short. So the question then is, what should we tell our students? What should we tell the public? Everybody in the world hears the word species. We have the Endangered Species Act. You open a flora or any field guide, you're going to see the names of species. We did a count in the introductory biology textbook, Campbell. The word species appears 2,400 times. Species are being used all the time. What do we tell our students, and what do we tell the public about species? I suspect that we kind of brush it under the carpet a little bit. We kind of imply that we know sort of what they are, or, I want to argue that we should be much more upfront about this. I want to suggest that we shouldn't just hide these difficulties. We should be open at the very least. You might say, I want to say we should teach the controversy. We should say, look, there is disagreement. There are people who really don't think species are real things. There are people who think they are. People have different views as to how we should approach a species problem. You should know that. And don't take species as units in the same way that you might take atoms as units or molecules. They are different. I would even go one step further and say that we should challenge our students, and I think we should address this head on and actually have defined for ourselves concrete and ambitious learning goals. So here are some that I would try and propose. I think it's important that our students, even introductory biology students, be able to define taxa and justify our efforts to name them, discover, to name them and systematize them, to understand that. I think they should be able to summarize problems with defining species as a special rank. I think they should be able to list phenomena that set the limits of who actually interbreeds with whom. They should talk about geography, mating potential, and so forth. And I think they should describe some of the factors that might affect the evolution of interbreeding potential. If we do all that, I think we're going to have much better students than if we just brush the concept of species under the carpet all the time. So the way that I've come to think about this, and sort of my summary here, is that we should let the public in on what I call biology's dirty little secret. So I found a book, it's a romance novel called Dirty Little Secret, and edited it a little bit. Because I do think that at heart, many of us know this is not a tidy concept. We want to believe there's something real, and I think we kid ourselves or we kid the public into believing that we know what we're talking about when we use the word species. I think that nested historical groups can be detected, and they're absolutely important to study, but I don't think any of them are species entities. I personally believe that selection on breeding potential can be studied, it's very important to study, but again, they don't integrate species in any meaningful way. So I will leave it there and say thank you for your attention. I hope I've generated at least some discussion, and I'm happy to take questions. Thank you. And if you do want to ask questions, there are microphones, number 1 and number 2. And it might be hard for me to see, but okay. Hi.
Hi, very interesting talk, thank you. I guess at the graduate level, I'd be in favor of teaching the controversy, as you say. To the public, I wonder, my main concern is that you brought up the Endangered Species Act, and so much of conservation and convincing the public that conservation is important is all about saving endangered species. And if we start saying, well, species aren't really a thing, does that undermine conservation?
I am totally sympathetic with that view, and I know that I've slipped into that way of thinking, and it's very defensible. I guess what I would say is it feels disingenuous to pretend something that we don't deeply believe. I think we should be more honest and talk about the fact that we want to preserve taxa. We want to preserve these historical groups. In the same way there's a tragedy if a historical work of art is destroyed, losing a taxon, this is a piece of history, a unique part of history, that is what we care about. And the fact that we might call some of them species or not is not really what I think we should be focusing on. We should be focusing on his taxa in general at all ranks, I guess. I don't know if that answers your question. I don't know what the next one was, but...
Hello. Hello.
Hi.
Thank you very much for this talk. I enjoyed it in many different ways that I'm not going to elaborate on for the sake of time. My question is related to your first part of the talk. These two criteria that you developed for to define a species, and you mentioned that they sometimes do not match. Is there a pattern for this non-matching? And for example, can you relate that to the way species form, to ecological versus mutational-driven speciation, for example?
Thank you. It's a good question. So the way that they will differ is if you take a gene that is, that's different from the background, the question is how far apart is common ancestry? So the differences will emerge depending upon whether the donor, sort of the source of the other history, is from a close relative or a distant relative. And so if all of the discordant signals have a similar depth history, then they won't be very different. But they can be different if you have cases where one group is receiving a lot of horizontal gene transfer from close relatives, another one is receiving them from very distant relatives. That's when they can start disagreeing with one another. I don't think it tells you much about speciation or that as such in the traditional sense, because it's primarily going to be driven by things like horizontal gene transfer. I guess over here, Rich.
Thanks, Dave. It was a great talk, very provocative, and I enjoy that. Your model that was looking at the fitnesses for hybrids. One thing that I'm wondering about, though, is let's just say that 99% of the genome behaves the way that you've shown, and you could just find that the mean is going up a peak, and everything is fine. What if they're just two genes that are like, Jablonski and Muller incompatible, that their contribution to fitness is such that they're sterile or dead, and the rest of the genes are no longer relevant to the fitness of the organism?
I guess I probably, because we will probably have to sit down and talk this through in a bit more detail for me to really make sure I'm answering the question. But if you're asking the question, we have two groups that do have Jablonski-Miller incompatibilities, then they do, so they're already sort of separate groups. I guess what I was talking about is sort of the arrow of selection from each point in time. If they are isolated, then they can stay isolated, clearly. But if we allow that there is some non-zero probability of breaking out of that, then the arrows of selection don't seem to be uniform to me within that space. And as a result, I don't think we can see it as a situation where the selection on interbreeding is sort of a cohesive force that's pulling the individuals into unity and preventing outbreeding. If they're not outbreeding, then clearly they can't break out of that. But if they can, then it's not obvious. It's not sufficient, I don't think, to just have the an incompatibility mechanism segregating in the population. I'm not sure if I got it. We should probably talk about it. But thank you.
Hi, David. Great talk. I'm a little bit worried about the non-existence of species, given that many theories that we have in macroevolution, even in community ecology, will crumble without the existence of species, right? So I wonder if it's a matter of we just don't know how to discover species, how to delimit species, instead of that they don't exist.
So, thank you for, I think that there is a sense that we built a lot of theory that uses the notion of species. But if we don't really know what species are, then what have we built it on? So, I think that, by the way, that Madison and Witten paper, they do talk about diversification rate studies, if that's one area that, and how that would be affected by this uncertainty. But it feels to me just sort of, it's not good enough to say we built theory on this concept, this theory is useful, therefore the concept must be valid. It feels to me we have to say, no, what is this unit that we're building it on? If we're not building on a solid foundation, I have skepticism about the resulting theory, however useful it may seem to be. We'd have to find out, well, what is it really doing? And does it depend upon there being a cohesive entity with a distinctive rank or not? And I don't think it can be, because I have come to believe that those things don't exist.
Yeah, my impression is that we have tried for centuries to try to define species in one sentence or two.
Yes.
And we need to realize that if they exist, they are more complex than that, and they may need a theory rather than a simple definition or a concept, what we call it.
Yeah, I suppose there is this view that some people have, it's sort of the ostensive definition. We perceive things, but we perceive things that aren't real. We perceive constellations. So we've got to be careful about inferring that because we perceive these entities, if we just work hard enough, we could figure out what defines them as a general category rather than this particular thing. We can look at a particular species and say, this is a group because of history, or because it really is got limited, the gene flow is limited to this set of organisms. But I don't know about the general category. Yeah. Who's that? Hi.
Yes, good morning. Thank you for your honesty about about that concept, about that situation for species. My name is Pani Dikosod. What I'm going to suggest is something very provocative, and I've presented a talk about it in the virtual component of this conference. It's basically, what do we think about thinking about the real unit of life, not really species, but lineages. And we would consider species as temporal configuration of a lineage. So you would take the human lineage, if you take the lion, Panthera leo lineage, or the polar bear, Ursus maritimus lineage, and we would consider the species as temporal configuration of each lineage. Let's say if you take the human lineage, Homo sapiens would be the actual configuration of that lineage. At some point, the configuration was Homo erectus, and at some other point in the past, it was Australopithecus afarensis. That's a very provocative concept. It's against the grain of the paradigm now in evolution, in biology. There would be no bifurcation in evolution. There would be no bifurcation, no divergences. Its lineage going all the way back, and the species would be just temporal, just ephemeral, whatever the current configuration of that lineage is. The lineage would be key, and species would be something different the way I presented it. So what do you think about it? So
Yes. I mean, so there has been this sort of movement for a long time in sort of a lineage concept, and it's been given different names. It's called an evolutionary species concept, or the general lineage species concept. I guess my main proposal is to say, think about a lineage and ask the question. You mentioned bears. I think it's hard to come up with a criterion of lineage where you don't have lineages within lineages. And so I think the idea that lineage defines a rank is going to be problematic, just as it is with concepts that look at relatedness. And then we can try and do what I said and to find some cohesive process. I just think we haven't found a cohesive process that says this is a lineage and therefore it's functioning in the world as a unit. So I guess we probably would have to sort of agree to disagree, and that's one of the things that I think is important to note is that I'm saying we should teach the controversy. I'm not saying we should teach that species aren't real. We should just explain why some people don't think that they are real entities. But yeah, thank you. So I think we should leave it there. I should let people go get a good break before the ongoing meeting. So thank you for coming and for supporting systematic biologists, the SSB, and come to the business meeting today, please.